Yeah, it's really a pleasure to be here today to present to you the Organoid platform. So before I start, I want to emphasize that I do this on behalf of all the partners within the Organoid platform. So I'm representing here the Princess Máxima Center and leading the Organoid platform. But within the platform, we have several other partners as well, from UMCU, Onno Kranenburg, from the Utrecht Institute, Johan van Es, and two companies, Mimetas and Xilis. So the problem. So we think that one of the reasons that findings that we do in the lab and the inefficiency of these findings to end up in the clinic is caused by the fact that we don't have good preclinical models that are representative of patient tumors. We are dealing with a very heterogeneous patient population, many different tumor entities, but even within the same tumor entity, every patient tumor is different. And to make it even more complicated, within a tumor, every individual tumor cell is different. So we need models that are representative of this patient and tumor heterogeneity, this intra- and intertumoral heterogeneity. That is really representative of this very heterogeneous patient population. So as I mentioned before, we think that one of the reasons that there is such an inefficient translation of preclinical findings to the clinic because of the fact that we don't really have these representative cell models. Of course, the best model is the patient. Well, we cannot use patients in the lab. So we have to make use of other models. And historically, people have been using cancer cell lines. And I think it has become more and more clear that these cancer cell lines are very poor representatives of patient tumors. They are grown in 2D, very artificial. They have been grown for decades in the lab and do not resemble anymore genetically or phenotypically the tissue from which they were originally derived. We can make use of animal models. There are advantages, disadvantages. But a mouse, for instance, is not a human. So also there are very clear differences physiologically, but also disease progression-wise that make animal models not the perfect model. So within the Organoid platform, the solution that we would like to provide is more representative human cell models. So we go beyond just organoids. We also make use of other representative models. And one of the take-home messages that I want to mention today is that no model is perfect. Every model has its advantages and disadvantages. And I think as a researcher, if you are aware of the fact of the advantages, as well as the disadvantages of the models that you're using, every model has its value. But we are convinced that organoids are more representative of patient tumors than classical cancer cell lines. But again, and I will come back to that later, also the models that we want to use within the Organoid platform have their disadvantages as any model. So the models that we use and provide and make available within the Organoid platform are obviously organoids, but also microorganosphere technology, which is provided by Xilis, a company that is involved in Organoid platform, and Mimetas with organoids on a chip. And I will go into these different models step by step. Organoids. So I will very briefly introduce organoid technology for those of you who do not know. And of course, organoid technology, adult stem cell-derived organoid technology, was pioneered in Utrecht, in the Utrecht Institute in Utrecht by the lab of Hans Klevers. And it all started with the discovery of the intestinal stem cell and a marker of the intestinal stem cell called LGR5. It's a cell surface marker involved in Wnt signaling, which was a very well-known pathway involved in intestinal regeneration. So Nick Barker, a postdoc in the lab of Hans, identified LGR5 and made a mouse model where he knocked in GFP, so a green fluorophore, in the endogenous LGR5 gene. As a consequence, all LGR5 positive intestinal stem cells are green, so they light up green, so you can see them. He could show that these stem cells generate all the differentiated progeny that is present in the intestine, and they are long-lived, so having all the features of a stem cell. One important feature of this cell was that it was proliferating, so it was not quiescent. Back then, people thought that stem cells were quiescent and would only start proliferating upon injury. But one of the observations that they made is that this intestinal stem cell is highly proliferative. So the question that they asked then is, can we take these intestinal stem cells, can we isolate them from the intestine, and can we grow them in a plastic dish? So Toshiro Sato, another postdoc in Hans' lab, used the mouse model that Nick had generated, and he purified these green cells from the intestines of the mouse. He put them in a plastic dish, he embedded them in a gel called matrigel or basement membrane extract, providing the extracellular matrix that epithelial cells need to grow and to remain viable, and topped it up with medium containing growth factors that were known to be required for self-renewal of the intestinal stem cell. These are Wnt, EGF, noggin, and arspondin. Wnt and arspondin activate the Wnt signaling pathway. EGF receptor signaling, and noggin is an inhibitor of BMP signaling, which normally induces differentiation. That is not what you want, because if they differentiate, they stop. So you want to keep them to proliferate, and you can do that by adding an antagonist of BMP signaling, which is noggin. And what they observed is that after approximately 2 weeks after plating these single cells had developed into multicellular structures composed of hundreds, sometimes thousands of cells. And the fact that they were grown out of this mouse model, where this green fluorophore was inserted, also allowed them to visualize the stem cells in these multicellular budding structures. And what they observed is that these GFP green cells, these GFP positive green cells, are residing at the bottom of these budding structures within these multicellular structures. And very nicely representing the in vivo architecture of the intestine with crypt and villus domains. So there's very nice organization. You can go from one cell to hundreds of cells within a couple of weeks. So it's a very nice way to expand tissue very efficiently, while they maintain the phenotypic features of the tissues that they were derived from. And because there is organization, they call them organoids. Mini guts, mini intestines, there are all kinds of names. But the most used term for these structures is indeed organoids. So since then, organoid technology has been developed for many other organs, including the lungs. And also here there's a very nice example at the top right, a lung organoid. So it's a balloon-like structure in this case. So it's cystic, so it's empty on the inside. And you have this single-layered epithelium. And what you see this ball spinning, that's actually mucus. So this mucus, so the slime is produced by cells in this organoid. And it's spinning because you have ciliated cells, which are also present in your lungs. Showing that indeed also here we see the different cell types that are also present in normal lungs in this lung organoid. So a very important feature of organoid technology is that you can develop these cultures with a very high efficiency from patient-derived tissues, healthy, as I showed you 2 examples, but also diseased, like cancer tissue. I always compare it to cancer cell lines. So if we receive 100 pieces of tumor, and we try to establish an organoid, we will succeed in 70–80% of the cases. If we try to establish a cancer cell line, we will only succeed in, 5, for lucky, 10 of the cases, so it's much less efficient. And if we plate 100,000 tumor cells, then using organoid technology, 50,000 cells grow out. While with cancer cell lines, it's usually a clonal expansion, so it's only 1 or 2 or 3 cells that are able to adapt to these artificial culture conditions. So you completely lose the inter- and intratumoral heterogeneity when you establish a cancer cell line, while with organoid technology, this heterogeneity is much better retained. Still not perfect, but it's much better retained. Yeah, as I already mentioned, they retain the phenotypic features of the tissues that they were derived from, but many studies have shown that also genetically, they are very much resembling the tissues from which they were derived. And one of the first examples was where they generated an organoid biobank for colorectal cancer. One of my former colleagues, Mark van Wetering from the Cleves Lab, published this paper in 2015, where they obtained colorectal cancer tissue and as well as adjacent histologically normally appearing intestinal tissue. Using organoid technology, generate organoids from both the tumor and patient-matched healthy tissues. Do DNA sequencing, RNA sequencing, drug screens to see, do these tumors genetically and phenotypically resemble the tissue from which they were derived? And if we do drug screens, can we find new drugs? So these H&E stainings, I will not go into those in too much detail, but the message here is that phenotypically and histologically, the organoids resemble the tissues from which they were derived. So because we can establish these models with such high efficiency and heterogeneity is so well retained, this gives potentially the option to develop individualized therapies. Many studies have shown that adult cancer-derived organoids have predicted value for drug sensitivity. So several studies have treated the organoids with the same drugs as the patients were receiving, showing that indeed the drug sensitivity of the organoids mimicked the drug response of the tumor in the patient. So this is a nice animation that I got from Hans, showing how we ultimately envision how organoid technology could be used in development of individualized therapies. So in the movie, this is the image of healthy organoid. So this is a patient with colorectal cancer. The different organs, from these organs we can grow organoids. In this case, this patient has a tumor in the colon. During surgery, the tumor is removed, and of course also healthy margins are taken along, so we can obtain both healthy and tissue and tumor tissue. We can grow organoids from both. It gives us the opportunity to, from very limited input material, expand it to large amounts of tissues, which then gives us the opportunity to test many different drugs. The tumor organoids typically contain a particular genetic alteration, which made it develop into a tumor. Because we can expand the tissue, we can test many drugs, and because we can also do that on healthy tissue-derived organoids, we aim to identify drugs that selectively kill tumor cells and not healthy cells. So in that way, develop less toxic therapies that does not harm healthy tissue. So what do we do within the Organoid platform? So first of all, we generate fully molecularly characterized patient-derived tumor organoid biobanks. So we selected several tumor entities, also together with the cohorts, so that we can very nicely couple our models also to the clinical information of the patient. Amongst others, we selected colorectal cancer, bladder cancer, head and neck cancer, and several pediatric cancer entities. We will perform whole genome sequencing, RNA sequencing, as well as histological characterization to ensure that the models that we have derived mimic the tissues from the patient. Again, as I said, it will be coupled to the clinical information within the cohort platform, and these models will be made accessible for third parties, collaboration with academia, but also commercial partners. Another very important task that we are going to perform within our organoid platform is to further develop the technology. Organoid technology now allows us to grow tumor cells, but the tumors are not just composed out of tumor cells, but also healthy cells, the tumor microenvironment. So we're also trying to develop co-cultures between these tumor organoids and different TME components, like, for instance, immune cells, but also stromal cells, but also the microbiome, to identify, for instance, bacteria that could be carcinogenic, to really make these models even more representative of patient tumors than they already are. As already mentioned, within the Organoid platform, we are not only developing organoids, also other models that we think could be very well used for different types of cancer research. And one example is the MOS technology, microorganospheres, very similar to organoids. Main difference is that it's a microfluidic approach, so they make use of very, very tiny matrigel or BME droplets. And the advantage of that is that all cells within this droplet are exposed to the growth factors that are in the medium. And in this way, you can generate short-term cultures that are much better retaining the tumor microenvironment. So this is an example. So what they can show here is that over a period of 3 weeks, in MOS, they can maintain tumor cells, but also different types of immune cells. And they're currently studying how much they do change in their cultures and see how much they are still reflecting immune cells in actual tumors. But this could be a potential solution for the fact that we lack immune cells in our organoids by using this MOS technology and thereby maintaining the tumor microenvironment. So this is a technology that is provided by Xilis, but then we also have mimetas. Several of you will probably have heard about mimetas before. So they are famous for their organoplates, organs on a chip, organoids on a chip. So they have generated these organoplates for different types of tissue, and within the Oncode Accelerator Organoid platform, we also want to include tumor cells, so that we can also look at communication between tumor cells and different types of healthy tissue, but for instance also make metastasis on a chip, where we can, with microfluidics approaches, try to mimic a seed and soil hypothesis and see how we can model metastasis on a chip instead of using mouse models, where we are fully dependent on now if we want to study metastasis. So they've developed these organs on a chip for different types of healthy tissues already. Here are a couple of examples: liver, nervous system, lung, intestine, kidney, and they are continuously developing this further. And again, as I mentioned before, is that within the organoid platform, we want to include the tumor tissues there as well. So with that, I've covered everything that we're doing within the organoid platform. We're very open to collaborate with anyone. So if you're interested or have any additional questions, please let me know. Here's my email address, and I can always connect you to other partners within the platform. For instance, one of the companies that you might be interested in collaborating with. And with that, I'd like to thank you and happy to take questions.